Hello and welcome to this Bio43 Pre-Lab video. Today we're going to talk about t-tests. This is the statistical test to use when you have a categorical independent variable and a continuous dependent variable, and you only have two treatments. During last week's video on bar graphs, we were discussing just such a scenario. We hypothesized that, because growth media has a higher concentration of nutrients than lake water, lab-grown tetrahymena are larger than tetrahymena found in nature. We set up an experiment where we used a microscope to measure the lengths of 10 tetrahymena cells from fake lake and 10 from a lab stock. We identified our null and alternative hypotheses, checked over the data for structure and quality, got our summary statistics, and plotted our data. We can see there's a difference between the two conditions, but we don't know if the difference is significant. Is it due to the independent variable, or just random fluctuations? After the last video, you know that the way to answer this question is with a statistical test, a measure of the signal-to-noise ratio, which will allow us to find a p-value. For that, we're going to use Vassar-Stats. Just vassar-stats.net. There will also be a link on our Sakai Pages toolbar. Click on t-tests and procedures on the left here. Two-sample t-test for independent or correlated samples. We'll always use independent samples, so click there. Then just copy and paste your original data into the two sample columns here, and click Calculate. All the numbers we care about will be in the first two tables. You can ignore everything below that. The first box is just the data summary. Check these numbers to make sure you copied the data correctly. The end value is 10 for both, which is correct. The means match what we calculated in Excel. Looks good. The second table has the key stat values you'll want to record. Here's the t statistic. The sign is arbitrary. You can just record the absolute value. Also note the degrees of freedom here. And finally, there is your p-value. There are one-tailed and two-tailed values listed here. Which one you choose depends on how you formed your hypothesis. If you thought the independent variable would have an effect on the dependent variable, but didn't give a direction for the effect, go with two-tailed. If you did specify a direction, use one-tailed. To take our current situation as an example, if you just said the source of tetrahymena has an effect on their size, you choose the two-tailed. But if you specified that lab-grown tetrahymena are larger than wild tetrahymena, pick the one-tailed value. Most of the time, we will use hypotheses that specify a direction of effect and therefore use the one-tailed p-value. So we have a p-value of about 0.002. That's well below our threshold of 0.05. So we reject the null hypothesis, and this supports our alternative hypothesis that lab-grown tetrahymena are larger than tetrahymena found in nature because lab growth media has more nutrients than lake water. However, due to a variety of confounding variables, it's really difficult to say whether the mechanism we provided is really responsible for the larger growth. Let's address that in the next video. See you then.